And I'm going to record the lecture. So apparently the lecture I did on that wasn't recorded and wasn't uploaded. Is that the case? So here will be a recorded example for you that you can use. So again, a review of metabolism, cellular respiration. So metabolism is a twofold process. Number one is anabolism, or building up. The second is catabolism, or breaking down. Anabolism starts small, uses energy, and builds. It starts small, builds to big, and it requires energy to go in. It's like building a brick wall. You've got to haul the bricks in, you've got to lay the brick, you've got to stack the brick, you've got to mortar the brick. It's an energy-intensive process. See a couple of you nodding. You done that before? I have too. Not something I recommend. Catabolism is the opposite. It is tearing down. You go from big to small. It is exergonic, meaning it releases energy. On an exam, I might ask you to define metabolism, and you could list both of those. That would be a good definition. You could say it's the sum total of all chemical reactions. That's another good definition. Could say it's breaking down the big to release energy, building up the small to absorb or store energy. Put it in your own words. Just show me you've got the concept. Is that clear enough for everybody? Any questions? Yes, sir.
Question not related to this. Have you graded the exams that we took?
I'll talk about that in the class. I've got most of them done, and I'll post them tomorrow. So, everybody feel comfortable with metabolism as a whole? We're not going to do an example of every single type of biochemical pathway there is out there. It's just too complicated. Is everybody okay with that? The only one we are going to look at is cellular respiration. The sum total of this, I put that equation on the board, but you may want to remember it yourself if you haven't written it down already. You start with sugar in the form of glucose, C6H12O6. You combine that with 6 molecules of oxygen, 6 O2s. So there are the two things you have to start the process with. You have to have your fuel, that's your sugar. You have to have the oxygen. The oxygen is ultimately going to be an electron acceptor. It wants electrons. We are going to strip them off of the sugar. Is everyone clear on that? We are going to end up producing 6 carbon dioxide molecules, CO2. The carbon dioxide is the waste product. It is what we have broken the sugar down into. Does everyone see that? We start with one big molecule. It has how many carbons? 6 atoms, right? We break it down into 6 individual single carbon molecules, right? 6 CO2s. We breathe those out, we're done, right? Wrong. We also produce water, H2O. Dihydrogen monoxide. Bet you've never heard it called that, have you? And that's it. We get about 32 ATPs from this. Now remember, ATP is the energy source for the cell. It is used by the cell for everything. Every single chemical reaction, every single physical action, every single building of a molecule. Even to start the process of glycolysis, you've got to have ATP, don't you? So it is what we want. We want lots and lots and lots of ATP. We're going to get about 32 of them out of this. We have to put in 2 to start, but we gain a total of 32 at the end. It's a pretty good investment, isn't it? 16-fold return? I don't know anybody would turn that down. It is the only way the cell gets enough energy, is to be able to use this process. Oxygen is not optional. No oxygen, no respiration. No respiration, no life, period. It's not a maybe, it's a definite. You're dead. 3 minutes max. It's not something I recommend. Been there, done that. 0 of 5 stars. Gave it a bad review on Yelp. Yeah, I know it's Tuesday, but it feels like Monday. We're all dead today. This is my 3rd cup. Plus I had a double shot of espresso. I needed it. So everybody with me? Now I want you to know that the central part of this is the citric acid cycle, or the Krebs cycle. It is the center of all these different pathways that feed into it. It is at the middle. The citric acid cycle has several steps in it, but it always comes back to the same start point. That's why we call it a cycle. Where we feed into it, there are different paths leading in. The example we're going to look at is glycolysis. Glyco is a root meaning sugar. Lysis comes from a Greek word meaning to separate, to break. So we are going to be breaking open a sugar molecule. Is everybody with me right now? We all on board the bus. So the first of this 4-part process is called glycolysis. The first step is called glycolysis. And you've got to put into it 2 ATPs to start it. And then you go through 9 separate chemical reactions. No, we're not going to look at them. No, I'm not going to draw them. I'm not going to tell you what's involved. I'm not going to tell you the structures. This isn't biochemistry. Be grateful. And before you ask, yes, in biochemistry, you had to learn the chemical formulas of all of these. You had to know the name of the enzyme. You had to be able to differentiate which one of these occurred the fastest and what factors affected it.
I'm guessing that's a test question as well.
No. This will not be a test question to list how many steps there are. I'm just telling you.
Oh man, I get biochemist.
Oh yeah, in biochemistry, I was evil. People hated taking me for that, which is fine. I hated teaching it. Then I became department chair, and I made sure I didn't assign myself to that course anymore. So is everybody with me now? Basically, what we're doing is two things. The first is we're going to rearrange the carbon atoms. Be like rearranging the furniture. If I wanted to have a clear path to march back and forth right here in the center, I would need to have some of you move some chairs, wouldn't I? That's all the rearrangements are, to make it easier to ultimately snap or break that 6 carbon molecule into 3 carbon molecules. Does everybody see that? And then we rearrange those further into our final form. I get done with my class, I get done with the pacing, I have you all bring all the chairs back so that it's ready for the next class. Is that understandable so far? Everybody okay with that? Thumbs up if you are. Hey, most of you are with me. COVID is what got me used to doing that. I had to teach, not on Teams, but what was the other one? Zoom. And apparently I didn't realize that was the like button. I told my students to give me the thumbs up if they got it button. When I'm asked me why I always had them like everything I said. Technology. So now we are done with the first part. We had to put in how many ATPs?
2.
We got out a total of 4. You put in 2 quarters, you got 4 back. Pretty good deal, isn't it? Net gain is 2. That's not a lot. We want 32 before we're said and done. Okay. All of this took place in the cytoplasm of the cell. It's floating out there in that jelly-like, syrup-like gunk. And we end up with something called pyruvate. P-Y-R-U-V-A-T-E. Pyruvate. Is everybody with me?
Is that for the second formula?
No, that's what we ended up with. We went through glycolysis and now we are at pyruvate. Everybody okay with that? We are going to move this guy into the center of the mitochondrion, into that space we called the matrix, the mitochondrial matrix. In the process, we are going to strip off 1 carbon dioxide, just 1. We are going to combine this with another molecule called oxaloacetate. O-X-A-L-O-A-C-E-T-A-T-E. Everybody okay with that? This combined molecule is called citrate, C-I-T-R-A-T-E. And now you know where the citric acid cycle gets its name, right? And we are going to burn it through another 8 steps. We started with how many carbons?
6.
We split it off into pyruvate, didn't we? Everybody remember that? Before we started the citric acid cycle, we pulled one off as CO2. Everybody remember that? So we're going to turn the cycle twice because we have two carbon molecules now, don't we? What we are doing during these next 8 steps is simple. We're getting 1 ATP. That's not a lot. Now that's 1 per turn, so we're going to turn the circle twice, right? So we ended up netting 2 more ATPs. We are going to reduce that 2 carbon molecule to 2 1 carbon molecules of CO2. We're done breaking that sugar molecule up. Does everybody agree? Then we are going to regenerate our oxaloacetate. It basically is like a truck that drives the same stretch up and down, or going in a circle. You load it, it carries it around, makes several stops, it gets unloaded. By the time it gets back, it's freshly unloaded, but it's still the same truck. Does everybody see how that works? It's ready for some more pyruvate to come on in and make citrate and keep turning the cycle. Does everyone get that? Thumbs up if you do. Okay. Good. We're done with the hardest part. The last thing we are getting out of this are a bunch of electrons. We are stockpiling those things. Y'all are probably too young to remember arcades, aren't you? Coin-operated video games, do they still have those? That shocks me. I didn't think they were allowed anymore. But back in the day when you knew you were going to the arcade at the end of the week, you wanted to save up a bag of quarters because back then that would buy you a play. I don't know what it is now. You probably wave your cell phone at it and it does something magic.
It's a card now. It's like you buy a card and just scan it.
You buy a card. Well, you get the idea though. You wanted to stockpile them. So throughout the week, any time you got some change, you threw it in there. So the next time you go to the arcade, you've got a big stockpile of quarters. That's what we're doing with our electrons. We want those electrons. Is everybody okay with that? You all with me? Those electrons are the equivalent of a slinky at the top of stairs. You ever play with slinkies? Those little coils, you just tip them over and they walk down the steps. That's because they are losing energy. They're going from a high energy state to a low energy state. We are going to pass them, this is step 3, it's called electron transport. We are going to hand those electrons off in a chain of molecules. One will take it, hand it to the next. One will take it, hand it to the next. But we're going down an energy curve. We're going down an energy slope. Does everyone get that? That allows us to harvest the energy. Every time those electrons fall down to another molecule, we use that energy to move a proton from the matrix to the space between the membranes. From the matrix to the space between the membranes. Does everybody understand that? So we have taken our stockpile of electrons and we have exchanged it for a stockpile of protons that we have moved into this space between the membranes. That's a high energy concentration, isn't it? It's going to want to come back inside, isn't it? And that's exactly what it's going to do. It's going to come back through a little protein that's kind of like a wheel. That protein is called ATP synthase. And this is the fourth stage of cellular respiration, making ATP. Every time a proton passes through, that energy is used to turn the wheel, grab an ADP and a phosphate and slap them together to make ATP. And we're done. You got that?
I think so.
You got that?
I got most of it as much as I could.
Does anybody need me to go over this any further? Does anybody have any questions at all about this whole muddled process? Yes, sir.
Okay, I got 1, 2, 3. What was step 2?
Step 2 is the citric acid cycle. So step 1 is glycolysis. Step 2 is the citric acid cycle. Step 3 is electron transport. Step 4 is ATP synthesis. We started with glucose. We are done with that. We've got a stockpile of ATP. We're happy. Rebecca, did that get you what you needed?
I think so. I'm gonna have to review it just because there's a lot of parts, but I definitely heard you.
Okay, and I did record it.
Yeah.
And I spoke loudly and I put it right in front of me.
Yeah, I just need to review it without having to write it down at the same time because it will make more sense.
Do my videos give you a transcript?
Yeah.
Yes.
Okay, I wanted to make sure I had that setting turned on. Things I do for you all. Back in the day, I had to make sure I had PowerPoint slides. Before that, I actually had an overhead projector.
You still do.
I still do.
Not that kind.
There is something over here that acts like an overhead projector. I wouldn't bet the farm on me getting that up and running. So, does anybody have any last things to say about cellular respiration, or can we put this in the past history bin? And there was much rejoicing, yay.
There was a little bit you did go over that day on photosynthesis, the beginning of it, that we didn't get recorded.
Okay, y'all want me to hit photosynthesis?
Just the beginning part.
2 parts to photosynthesis. This is essentially nothing more and nothing less than the reverse of cellular respiration. In this case, we start with water, CO2. We end up with oxygen as a waste product and sugar. The only real difference is instead of building up, or instead of breaking down a molecule to release energy, we are harvesting solar energy to make a big molecule.
It changes to chemical energy, doesn't it?
Yes, we harvest it and convert it to chemical energy that we can use. Whether you like yourself a nice sweet stack of pancakes, or whether you like yourself a nice Baconator from Wendy's on the way home, you get that energy eventually, don't you? This is the foundation for almost all the energy used in the entire biosphere. And the entire biosphere literally means every living thing on Earth. There are some out there in left field oddball exceptions. Things like purple sulfur bacteria. Things that live down near volcanic hot vents 10,000 feet deep in the ocean. We aren't going to worry about those. Is that okay? Most of the photosynthesis that sustains us actually is not done by plants. Instead, it is done in the oceans by something called phytoplankton. Phytoplankton. Phytoplanktonmous. Nobody gets the reference. Which is fine because I barely got it and I grew up in that era. Is everybody with me? You understand why this is an important process? You get two things from it, both of which are critical for life. No photosynthesis, no life. It's that simple. Number one is the energy that everything uses. Number two is the oxygen that we use for respiration. You all don't need me to review the anatomy of the leaf, do you? Okay, so again, make sure you understand that the leaf is nothing more than a big photosynthesis factory. That's why they are so thin. The only thing they want to do is grab sunlight and CO2. Am I going too slow, too fast, or just about right? Okay. So now we've got two separate types of reactions in photosynthesis. Huh?
Is it the light and dependent?
The light-dependent reactions, yes, those are the ones that require sunlight.
And they can happen in the dark. Can they?
Not the light-dependent. That's what dependent means. It has to have light.
Oh, it's in the
And then the light-independent reactions. Don't call them the dark reactions. We're not dealing with Darth Vader here. Every once in a while, one of your students will do something. I think they do it just to mess with your head, to see if you're paying attention to them. One of my students just stuck a pencil up their nose. Now everybody's looking to see who it was. I won't say, but I'm watching you. Back in the very early days of when we had Wi-Fi networks and all that, the students would bring in their laptops, sit on the back row, and play some Facebook game called Mafia Wars. One day I was writing something on the board, had my back completely turned. And I said, and you on the back row, I know you're playing Mafia Wars. Instead of paying attention to the lecture, I hear the slam, crash, clatter, clatter, thump. And I turned around and one young man had slammed his laptop shut, knocked it off the desk, reached over to try to grab it, and fallen out of his chair. And since that day at that school, I had a reputation of having eyes in the back of my head. It was kind of funny. So now we're back to it. The light-dependent reactions require what? Light. Gee, imagine that. They take place in the chloroplasts, in the stacks of thylakoids. These are basically going to do two things. Number one, they are going to split a water molecule. They're going to take the light they get and they are going to use that energy to split apart a water molecule. That water molecule is going to become three things. Number one, oxygen that is released. The oxygen is a waste product. We don't need it anymore. Number two, we're going to strip some electrons out. And number three, we're going to be left with some protons. Some protons. We are going to directly make a small amount of ATP. A small amount. As long as we have the water, we can split it, release the oxygen, and make some ATP. We are also building up a huge stockpile of electrons. Is everybody with me? As long as the sun is shining, we are going to continue with the light-dependent reactions. There is one problem with this process. We have to get rid of that oxygen, don't we? To do that, we have to open our stomata. Remember those things that look like little mouths on the bottom of the leaf? Those open up, speed into the spongy mesophyll, and allow for gas exchange. They allow for gas exchange. The oxygen goes out, but along with it goes some water. We lose some water. That process is called transpiration. Transpiration. And remember, as plants, we don't get to go down to the swimming hole and get a drink, do we? We only get what we can absorb through our roots. So we have a trade-off. We have a dilemma. What is the biggest threat to a plant? Drying out. The technical term is desiccation. We have to leave the stomata open to get the CO2 we need, to get rid of the O2 we don't need. But we also have to balance that with keeping our water inside. Which leads us to the second set of reactions, and these are called the light-independent reactions. They don't care if the lights are on or off. They happen. We need three things for this. Number one is ATP. Well, we just made a bunch of that while the sun was shining, didn't we? So we got that, don't we? The second thing we need are electrons. Well, we just made a bunch of those and stockpiled them while the sun was shining, didn't we? You see where I'm going with this? What we need now, the last part of the process, is the carbon dioxide to become the core of our sugar molecule. If we keep the stomata closed during the day while it's hot, we can open them at night and allow the CO2 in. If we use up all of the ATP and electrons we got throughout the day, we'll keep them open most of the night so we can stockpile it. Now, not every plant has to close them at night. Some of them keep them more or less open all the time. But it's an option that gives the plant a measure of control over the process. Everybody on board the bus? What happens next is called the light independent reactions, the Calvin cycle, sometimes carbon fixation, because we are fixing carbon out of the atmosphere. We are fixing it into a solid form. And when people talk about carbon releases in the atmosphere, what they are referring to is how much it has increased over time. It's almost doubled in about the last 30 or 40 years. Essentially, the Calvin cycle is similar to the Krebs cycle. It just turns a wheel. You add CO2 molecules to an increasingly large chain, and you eventually end with sugar. Now we are done. Thumbs up if you felt you understood it. Does any, yes ma'am.
One question.
Sure.
You're adding it to a chain of what?
An increasingly long carbon chain. An increasingly long carbon chain. You're just building a big molecule. Any other questions? Jillian?
Is the Calvin cycle part of the light independent, or is that like?
It's the same thing.
It's the same thing.
It's another name for it. My problem is I've been studying this for too long and I'm too old. Back when I learned it, it was the Calvin cycle and everybody knew it as such. Now we're supposed to get away from naming things after old guys, so I am an old guy, so I guess I'm stuck in the rut. But I apologize if that confused you. You can use any of those if you want to. If you talk about carbon fixation, the light-independent reactions, the Calvin cycle, and don't tell Case this, but even if you call them the dark reactions, I will still give you credit. Because this isn't a class for majors. And I want to make sure that everybody understands the whole process. Did that clarify it enough for you?
Yeah.
Rebecca, did this help you?
Yeah, it's fine. I just need it recorded because then I can look at it after. When you're writing to really get it all.
Well, the counter is still going up and the little red light is on. So apparently I did the right thing. I pushed the right button. This used to be so much easier. All you had to worry about was a piece of chalk and an eraser. Now you got to make sure that your Wi-Fi is connected to your phone while your tablet is on a different system, calling up the lecture while you... Back when I was in school, we didn't have no computers.
I bet it was a lot harder too.
Huh?
I bet it was a lot harder too, for sure.
Had to walk to school 20 miles each way, uphill. in the snow, barefoot, in July. I sound like a grumpy old man because I am one. The world keeps changing too fast for me.
It's bound to happen to everyone.
Huh?
It's bound to happen to everyone.
Well, I was telling you all about my mother at the start, but give her credit. She has not turned on the TV in about 3 months. But every night she comes in asking if she can borrow my old tablet, which sits on my recharger during the day for my use at night, because she's run all 3 of hers down. Granny's wired. She streams everything. I'm glad she enjoys it. So back to metabolism as a whole. Does everybody feel like you've got a good handle on this whole thing? The one thing that makes it possible is the transfer of electrons. Remember I talked about redox reactions? Oil rig, oxidation is lost, reduction is gained. They always occur in pairs. Something loses electrons, something gains them. Oxygen is going to be usually involved in the stripping and gaining of the electrons because it's so electronegative, it wants them. And hopefully that will be the last of the biochemistry, and we will never have to open this particular can of worms again until exam review next time. Does that sound good? A couple of you are nodding emphatically. Last chance for any questions. Speak now, forever hold your peace. I always like saying that at a wedding. Yes, I've officiated weddings. Like I said, I've had a very colorful career in my life. So back to what we were supposed to be talking about 40 or so minutes ago. Cancer. I believe that was the very last thing we mentioned on last Thursday. Cancer, the best single definition for it is uncontrolled cell division. Uncontrolled cell division. If you say uncontrolled cell growth, I'll probably give you credit, but I want you to understand it's not the cell getting bigger, it's getting more of them. Is everybody clear on that? There is a very, very complicated network of molecular switches in your cells. We talked about the process of differentiation. You remember that when we started the whole stem cell? It's cellular specialization, right? What do you want to be when you get out of here? A shrug.
Farmer.
A farmer. It requires a specific skill set to be a farmer, doesn't it? I'm a biologist. I do not have that specific skill set. I don't know when is the best time to plant crops. I don't know which one are the best ones to plant. I don't have any clue how to get crop insurance or any of these things. I wouldn't know where to sell what I grow. I wouldn't know how to harvest it or store it. I'd be willing to bet you probably don't know how to extract DNA and run a DNA sequencer. We each have different specializations, don't we? Different training. Does everyone see that? It's taken you a while to acquire those skills, hasn't it? It took me quite a few years of school to acquire the skill set I have. Cells are very much like that. Once you have invested time and energy into specializing, you want to stay specialized and functional. You don't want a nerve cell suddenly deciding it needs or should be a brain cell. You don't want a nerve cell becoming a muscle cell, do you? You want those cells to maintain their specific functions. I cannot emphasize to you how critical that is. The more specialized a cell, the less likely it is to divide. The whole thing we did with stem cells was about that. The more switches you set, the less potential you have to go backwards. We talked about Hayflick and his limit, didn't we? Now, a living organism needs to be able to have cell division occur. As an example, in your body, the lifespan of a red blood cell is about 3 to 4 months total. Did you realize that? On average, you replace just about every cell in your body every 7 years.
He did talk about that.
So you have to have some cell division occurring. When you're younger and you're developing, it's even more critical, isn't it? I love joking with my niece because that kid grows like a weed. She's taller than my mom now, taller than her own mom. She keeps this up, she'll be as tall as me and her dad. It's kind of scary. Yeah, and she's only, well, she'll be 12 next month. I hate to think what happens when she hits that second growth spurt. But she needs to grow, doesn't she? She eats a lot. I can't eat a lot because I quit growing up and I started growing out. You see where I'm going with this? The growth phase in life, you need active cell division. What about when you get a cut? You want to be able to repair your skin, don't you? You want to be able to heal a wound. You want to be able to recover from an illness. You see where I'm going with all this? For all of that, you have to have cell division. So cell division is inherently both necessary and a good thing to have. The problem comes with controlling it. I like to go a little bit faster on the road at times. It's nice to have that accelerator, isn't it? But by the time I get up to Burnside and I start to hit the red lights and the traffic, it's nice to have that other pedal to slow down, isn't it? That allows me to control the average speed of my vehicle. You see where I'm going with this? Just like the accelerator and the brake in a car, you have the ability to control the rates of cell division within your body. What happens when you lose that control? Is that a good thing? You don't want to be in your vehicle when it suddenly goes to the floor and stays there, do you? My little thing won't get up past 100, I'm sure, but I definitely don't want to be trying to drive through traffic all the way home at 100 miles an hour. Yes, ma'am.
So, like, people with dwarfism, what is that cell division or something wrong with them?
Yes, because what happens, I'm just going to give you one specific example, achondroplasia. It's a form of dwarfism that's caused by a tumor or growth on the pituitary gland. That's the main gland underneath the brain that secretes human growth hormone. Essentially, the tumor interferes with its function, so it does not continue secreting enough growth hormone. And in that case, it's like going a mile down the road instead of driving 50 miles to my house and just slamming on the brakes. That help? Yes, sir.
There was a person in the 1900s where he had dwarfism, but like after a while, like when he hit a certain age, he'd grow up like
8 or 9 feet tall. I think it was the tallest man in the world. I remember hearing about that.
Yeah, it was like.
Ended up dying of a heart attack in his 20s.
Yeah.
Y'all heard of Andre the Giant? No reason you would have. He was a wrestler, but he was also in that cute movie.
Princess Bride? Yeah.
Yeah, the Princess Bride. He was the giant. And you see how big he is. He had a tumor on his pituitary gland. And he died very young too. I think he was in his late 40s when he died. And those are the central controls. Those are the long-term body-wide controls. That's not what happens in cancer. In cancer, what happens is you get a mutation in one copy of a gene. Cancer operates on what we call the two-hit model. The two-hit model. Let's see if you all can guess this. Who in this room has the overall highest risk of cancer, all other things being equal? There's one person in here who's at significantly higher risk than everybody else. Now, I'm eliminating genetics and I'm eliminating environmental and diet, so we're not assuming anybody's a smoker or a tanner. Point that finger at me. Just because you're right, I have the highest risk of cancer. Why?
You're older.
Well, you're really getting on my good side because you're entirely correct both times. I have the highest risk because of my age. What, 19, 20,
21.
21? I'm 56. 2.5 times your age, 3 times? Let's round it up 3 times. Who's had 3 times as much opportunity to be exposed to radiation?
I went to the beach before, but
I used to work with radioactive material. Who's had 3 times as long to get exposed to hazardous chemicals? Cigarette smoke. Bad things in my diet from eating red meat. Sunlight. You see where I'm going with this? Let's say that when I was 21, I got mutation in a cell in my left forearm. That's my first hit. How long does that stay with me?
7 years.
Forever. So anytime over the last 35 years, I could have taken hit number 2. And in that case, it knocks out both copies of the gene. Now let's say this particular gene happens to be the brake pedal that stops cell division. Yes, ma'am.
So, because you were talking about radiation, the radiation poisoning in the factories, like the young girls, like in the early 1900s, was that just like a celled cancer then, or was it just completely different?
That's completely different. Y'all want to hear what radiation poisoning is? Radiation takes a variety of forms. Basically, it's named after Greek letters. Alpha emitters, you can block them with a sheet of paper. They're not that radioactive. Beta radiation, I worked with beta emitter, phosphorus 32. I worked behind a plexiglass shield about that thick. You put the Geiger counter behind it, tick, 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 tick. You put it on this side of it, it wouldn't tick at all. X-rays, ultraviolet radiation. But what killed the radium girls was something called gamma. What's the critter in the Marvel movies that has a bad temper and bulks up?
Rocket.
Bruce Banner.
That's his name. The Incredible Bulk. He bulks up. Yeah, I just love how all this muscle just appears magically out of nowhere, like he summons it from an alternate dimension. If Bruce Banner got exposed to that much gamma radiation, he would not turn into the Incredible Bulk. He would die a fairly horrible and lingering death over a period of about a week to 10 days, suffering in great pain. That's radiation poisoning, because they were painting radium onto watch dials. Radium is a gamma emitter. Then to clean their brush, they would lick it. So they now have radioactive paint gamma emitting in their mouth. You know what blocks gamma? Very little. I got exposed to gamma source one time. Y'all know what a herbarium is? It's like a storehouse for dried plants. They were cleaning out the herbarium at Vanderbilt, and one of the grad students found a radiation source. So he brings it over to our lab because he knows we have a Geiger counter so he can check it and see what it is. He brings that thing in there, and suddenly the Geiger counter starts going tick, 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 like a clock. I said, "What do you got, Gary?" He said, "Oh, this is what I found over in the herbarium." And I said, "Put that behind the shield." So he sticks it behind my plexiglass shield. The Geiger counter keeps right on going. Tick, 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 tick. And I said, "That's hot." He said, "Yeah." I said, "Stick it in the centrifuge." The centrifuge is a big wheel that spins something like 10 or 50,000 Gs. It's got steel walls this thick, has a hatch that thick, solid steel. So we put it in there and closed the hatch and set the Geiger counter on the lid. Tick, 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 tick. I said, "See ya, I'm going home." That was my soul dealing with a gamma source. Now imagine you've got that in your mouth. That high-speed radiation is zipping through your cells. It is killing those cells by destroying the DNA. Remember we talked about apoptosis? You can lose a layer of skin, can't you? Sunburn isn't pleasant. It's not the most comfortable thing, but it grows back. Can you do that with bone? Can you do that with blood? What happens is, depending on how your dose and how much you got of it, you're now literally the Walking Dead. Louis Slotin was an atomic scientist just after World War II who was working with what they call the Demon Core, and he got exposed to a full body massive dose of gamma. And basically, they told him, "The only thing we can do is give you time to put your affairs in order, get your parents down here to say goodbye." He was Canadian. They had to fly his parents down in. This was like 45 or 46. And he died 9 days later. Basically, it kills off the fast dividing cells first, those in your skin. Hair follicles. You lose your hair. You get the absolute worst case of diarrhea and nausea and vomiting than you can ever imagine, because all of the epithelial cells that are continually sloughed off and replaced in your intestinal lining are now dying and being passed out of your system. But other than that, you feel pretty good because you haven't had enough cells die yet to kill you. Then your blood cells start to die, and you don't get them replaced because there's no more available. You've killed all of the stem cells. So you get anemic and you start getting weak, and of course you're not eating anything. And eventually your systems start to shut down and collapse and you die. Is that way more detailed than you wanted?
No, I think that's cool.
I think one thing I was surprised about, there was a guy that's about to Hiroshima bombs. It's like, one was Hiroshima and one was
Nagasaki. There were people who survived both of them. Hiroshima survivors who went to Nagasaki and they got hit with the second bomb. We're not going to go into that because you get me off on something like that, it's another rabbit trail we'll never get off of. But the main difference there in radiation poisoning, the damage is so intense and so severe and so immediate that your cells don't have time to start dividing uncontrollably. They're just dying, and there is no replacement. It's one of the worst ways to go because after that initial bout of nausea and bleeding, you recover for a day or three to the point where you feel more or less normal. But you're dead. There's nothing that can save you at that point. It doesn't matter what they do. They can do a whole transplant and everything. You're going to die because too many of your cells will. How many copies of every gene do you have in your cells? 2. One from mom, one from dad. Those copies are called alleles, A-L-L-E-L-E. An allele is an alternate form of a gene. An alternate form of a gene. Imagine you're given a chance to go back in time 20 or 25 years and buy a first edition copy of that book about the boy wizard. What was the name of that series? Yeah, that one. And you decide you really want a first edition copy, and you teleport over to merry old England to get it. And you go looking through the bookstores for Harry Potter and the Sorcerer's Stone. Are you going to find it? Because that's not the title of the book. Did you know that? Us poor dumb Americans don't know what the Philosopher's Stone was. So they had to change it for us Americans. Even though we speak English, we spell things wrong according to them. We don't stick a U in color. We don't stick a U in armor. You see where I'm going with this? It's just different enough that if you're not used to reading Brit Lit, you might be tripped up by the odd little spellings and word usages. But you could still read the book and follow the plot and understand pretty much everything, couldn't you? That's what alleles are like. It would be like a British edition and an American edition. You put them side by side, there's just enough little tweaks in there to make you realize this is not what I normally read. And yes, I've read a lot of Brit Lit, so I understand. Does everybody understand that? It's the same basic information. It's even the same presentation and sequence. There's just a few little tweaks. Grammar, punctuation, spelling. You've heard of the Oxford comma, I'm sure. Let's eat, comma, grandma, versus let's eat, no comma, grandma. That makes a big difference. Yes, I'm a fan of the Oxford comma. That's what alleles are. But again, generally, one is not necessarily superior or better to another. Is everyone clear on that? So what happens is, when you get the first hit, you lose one functional copy of that particular gene. Does that doom you to cancer? Because you've got a second copy as a backup. What happens at 56 when you get zapped by the second hit? We just took the brakes off of your car and didn't tell you. Think I'm going to make it home safe if that happens? May not kill me, but it's certainly not going to have a good effect if I get up to the highway speeds, is it? Because I'm not going to be able to slow down. Does everybody see how this goes? And that essentially is what happens when you get the second hit. That leads to cancer. The genes that say don't divide get eliminated. The genes that say divide get locked into the on position. There's a variety of different cellular mechanisms. We're not going to get too hung up on them. But that is how cancer starts. You get that second hit, and there is no corrective mechanism left. You may have been born with one hit already. If you got a dysfunctional allele from one of your parents, they may have gifted you with a 50% increase in your cancer risk, depending on the allele and the type. Is that a guarantee you'll get it? Somebody like me who's played with chemicals and radiation and eaten all kinds of red meat and grew up with a father who smoked, do you think it's a guarantee that I'll get it? No, it's all a numbers game. Keep in mind, a 1 in a million shot does not mean if you do it a million times, it's guaranteed. This is why people play the lottery. This is why people go to casinos. They do not understand what statistics and probabilities mean. If you spend $1 million to buy $1 million $1 lottery tickets, you are not going to make $1 million. Everybody clear on that? Age is the number one risk factor, period. It just means you have a higher accumulated genetic load, what we call a genetic load. Who has the highest genetic load in this room? Point that finger again. Because you all are right. It's me. It still doesn't guarantee that I'll get cancer. It doesn't mean that you won't. Some of you, as young as you are, could develop it tomorrow. I may never get it. It's just a numbers game. I'm not going to ask how many of you buckle your seatbelt. I hadn't had a wreck in probably almost 30 years until a couple of years ago. Somebody pulled right out in front of me. I didn't have a chance of even touching my brake pedal before I plowed right into him. Totaled my truck. I didn't even have a bruise. That was the one time I needed my seatbelt, wasn't it? My office is in Stoner Hall. I like to leave from here rather than go back to my office when I'm done teaching this class. So I park out here. Moving from there to there, just because I've made it habit, I buckled my belt.
At least you got a new car for your mom. At least you got a new car for your mom for free.
Well, no, she gave it to me.
I know, that's what I meant.
Yeah, I got a new car for Mom, and then she gave it to me because she didn't like the fact that I talked to her. I didn't show her the mute button. She still won't drive that thing. She had to ride in it the other night coming back from the hospital, and I don't think she was too happy about that. I told her to wait on me while I went in the house and turned the lights on. So before I even get the garage door open, here she is stomping up the driveway. That's probably why she is 85 and still so functional. I hope I'm in half as good a shape as she is. So does everybody understand how this process starts, what it is? It's all due to random factors. Can you increase your odds of survival in a car wreck if you buckle your belt? It's not to say that I would have had an injury had I not had it buckled that day, right? It's just me playing the odds in my favor. That is all cancer is. It's just a game of numbers. If you lived a totally vegan lifestyle, you lived way out in the boonies, off the grid, you ate everything organic, you never touched red meat, you never interacted with modern society, you never got exposed to any kind of something, you wore a hat everywhere you went, and SPF 9000 sunscreen, and could you still die from it? Bingo. There's no guarantees here, folks. What I always hope that people get from this lecture or series of lectures is the take-home message. There's risks involved in everything. Be careful about what you pick to risk something on. At my age, honestly, I am not as worried about certain things as I would have been at your age. I'm not about to start smoking, and I'm not crazy. I don't get a Baconator every week, despite me joking with you all. The last Thursday after the day I had, you better believe I went by Wendy's when I got down there. It wasn't a Baconator, it was a single cheese, but still, I enjoyed that. And considering I was sitting with my mom in the hospital for 4 more hours after a 16-hour day, I told her, I said, you don't say a word. I'm eating it with malice aforethought, and I'm enjoying it right in front of you. But I haven't had one since. And that's the first one I've had in 2026. You see what I'm saying? So I don't want you going out thinking you're going to get radiation poisoning like the radium girls, which was a great question, by the way. I'm glad you asked that. You're not going to get skin cancer walking across the parking lot and getting the sun shining on you. But if you're smoking 3 packs of cigarettes a day and you're doing the tanning bed every other day for an hour at a time, that's probably not a low-risk lifestyle. Whether or not you want to keep that up is entirely up to you. So how do y'all feel? Comfortable with this? You understand the risk of cancer? You understand genetic load? You understand it's variable? Some people are just born with a higher one. Well, it looks like one last thing. The tests are almost graded. I should be posting them tomorrow morning sometime. So if you want to check on Blackboard around noon, hopefully they'll be up.
How do we do on this? Like if you guys
What did you have your hand up?
What's the average?
I can't tell you I get all of them graded.
Have they been good so far? Yeah.
Huh?
Have they been good so far?
For the most part, yeah. And don't forget, I'm going to let you all correct them. So if you got a 70 on it and you do the corrections, it'll bump it to an 85. If you got a 98 and you correct the one you missed, you get a 99.
Oh, that's great.
If you got a 50, you could still bump it to a 75. That sound okay? See y'all.